Hello, and welcome to MCAT Metabolism Part 3. Today, what we'll be covering is the pentose phosphate pathway and the electron transport chain. So if you're watching Part 3 and you've already seen Parts 1 and 2, we covered everything up to the citric acid cycle in Part 1, and in Part 2, we covered gluconeogenesis, fermentation and Cori cycle, and glycogenesis, glycogenolysis. That brings us to today. We'll be going over pentose phosphate pathway and the electron transport chain. If you have access to the handout, then you'll see that we also have lipogenesis, lipolysis, and the carnitine shuttle in the handout as well. However, we didn't get to these in yesterday's session, and I'm trying to keep the sessions in sync. So we'll start next week with lipogenesis and lipolysis of the carnitine shuttle, and we'll make our way over to beta oxidation and fatty acid synthesis. So this is what I anticipate covering next week. All right, so we'll start with our intro to metabolism. For anybody who hasn't seen any of these videos before or hasn't been to our session, my thoughts on metabolism is that it is one of the most difficult topics for MCAT students to master. But a thorough understanding of metabolism being this sort of high-level topic that connects lots and lots of different, more fundamental, sort of lower-level topics, including OChem reactions, reduction oxidation, enzyme classes, bioenergetics, macromolecules, cell structure, digestion, and more. Tips for metabolism: connecting enzyme names with substrate names and bond changes. So this is crucial. If you can connect the name of an enzyme that's catalyzing a given step in a metabolic process, and you can connect it to what was the substrate, what was the product, what were the bond that changed during the substrate to product reaction, that will only help you to reinforce what's going on in the steps, connect them into a larger understanding and a narrative understanding. Look for patterns. We'll be pointing out lots of patterns between not only different metabolic processes, but other cellular processes as well. Ask, why is this happening again, often? So it helps if you can ask why. I like to say, try to understand things like a child does. The child says, why? You give the answer. The child says, okay, but why? And then you give an answer to that. And eventually you've broken it down to the most fundamental topics. And a narrative understanding is superior to brute force memorization. It may be true that not all of the steps that we cover in metabolism will be equally tested on the MCAT. However, understanding it like a narrative, understanding metabolism as a narrative from start to finish, can only help you to make more connections, establish a broader framework, and have these things stick better and longer. And just a personal tip, get a whiteboard, some kind of whiteboard. It doesn't have to be like the huge kind of whiteboard that I have right here. It could be just a desktop whiteboard that will allow you to just erase and rewrite, so that you're getting muscle memory, you're connecting more neurons, and this can only help things stick better and longer. So for instance, you could take glycolysis or some metabolic process, erase all the intermediates, fill them back in, then erase all the enzyme names, fill them back in. If you can look at the name of the enzyme, you should be able to, after seeing the process a couple times, you should be able to predict what is the substrate and what is the product that this enzyme works on. Or if you see the substrate of the product, you should be able to predict what is the name of the enzyme that catalyzes this step. And these whiteboard idea will serve you well for both the MCAT and med school. Pencil and paper is fine too if you just want to do pencil and paper and erase there, or just write it down a bunch of times on paper. That can work as well. But I've always been a fan of whiteboards. So we'll be starting today with the pentose phosphate pathway. So what is the pentose phosphate pathway? What is it used for? If we look at the term pentose phosphate, it seems like this is a pathway that involves how many carbon sugars? 5 carbon sugars, good, and that have a phosphate on them. So this pathway is named for production of the 5 carbon sugar ribose 5-phosphate. What do we use ribose for?
DNA backbone.
DNA backbone. So DNA, RNA, right? And certain coenzymes, as we'll talk about today as well, are going to have ribose. There are two phases of the pentose phosphate pathway, the oxidative phase and the non-oxidative phase. And these will serve different processes. And you won't have to necessarily do the oxidative phase to do the non-oxidative phase, as we'll talk about. And likewise, you don't have to do the non-oxidative phase. If you're doing the non-oxidative phase, you could also bypass the oxidative phase. The oxidative phase is used for production of NADPH. Now, we've encountered NADH a lot throughout glycolysis citric acid cycle. So NADPH is going to be a very related molecule, the only difference being a phosphate. But it's not going to be used for, as we'll see, oxidative phosphorylation. NADPH will be used as an energy source for other reactions and other processes in metabolism. The non-oxidative phase is used for production of ribose 5-phosphate, which is, of course, a precursor for nucleotide synthesis as well as coenzymes. So those are the two parts of the pentose phosphate pathway, and those are what they are used for in the cell. In my experience, for the MCAT, it's generally accepted that you don't need to know structures for pentose phosphate pathway intermediates. And you don't need to know all the enzymes or all of the substrates as well, intermediates, only certain ones. So we'll be emphasizing the important enzymes to know for pentose phosphate pathway that you could actually be tested on, and the important intermediates as well. And whereas it's occurring, the location of the pentose phosphate pathway is in the cytosol. Any questions before we get started and go in detail?
Let's do it.
Let's go. All right, so here's a diagram from Jack Weston on the pentose phosphate pathway. I like this diagram because it is very nice and clean. It has most emphasized details present. So we have a very sort of bare bones skeleton sort of diagram for the pentose phosphate pathway here. And this includes all of the most high yield things that you could be tested on, at least in my opinion. So we can see that there's the oxidative phase and the non-oxidative phase. We're saying that the oxidative phase is irreversible. Now, the reason for the oxidative phase being irreversible, the main reason, at least functionally, is because if we're running the pentose phosphate pathway oxidative phase, we want to make NADPH. If we're running the non-oxidative phase, we're making ribose 5-phosphate. So because the purpose of running this pathway is for those, it's ran only in the specific times when you need either NADPH and or ribose 5-phosphate, we don't want this pathway to ever really go backwards. So that's functionally why it's an irreversible process. We'll talk about chemically why it's irreversible as well. And we said non-oxidative phase produces ribose 5-phosphate for nucleotide synthesis. And we can see here's our end of our oxidative phase, ends with ribulose 5-phosphate. And then here's our ribose 5-phosphate. And as you can see, eventually we can send intermediates back to glycolysis if we don't use ribose 5-phosphate for nucleotide synthesis. So any questions here?
Hi, I just have a question. So how in the body can we decide whether glucose 6-phosphate will undergo glycolysis or undergo pentose phosphate pathway?
So how does the body or how does the cell decide? Yeah, so glucose 6-phosphate, of course, is an intermediate in a lot of different metabolic processes. We encountered glucose 6-phosphate in glycolysis. We encountered it in gluconeogenesis, and we encountered it again in glycogenesis and glycogenolysis. So based on the cell's immediate needs is how the cell decides what to do with that glucose 6-phosphate. If the immediate need is to make ATP, we would just send glucose 6-phosphate down glycolysis. And if we're in aerobic conditions, then we would run the citric acid cycle in the electron transport chain. If we're in a liver and the liver is in response to glucagon or cortisol or epinephrine, then that means that we need to raise blood glucose. So we would then send our glucose 6-phosphate, we would turn it into glucose and then export the glucose into the bloodstream so that we can go feed the tissues. If the cell is deficient on NADPH, in other words, if we need to either make cholesterol, make fatty acids, or we need to reduce more glutathione, as we'll talk about as an antioxidant. If there are needs for NADPH, then glucose 6-phosphate will go into the pentose phosphate pathway. And as you can see here, step 1 glucose 6-phosphate dehydrogenase is the rate limiting step of the pentose phosphate pathway. Did that answer your question?
Yes, thank you.
Awesome. So here we have a slightly more complicated diagram of this process of pentose phosphate pathway. And we're going to go through most of this diagram in a decent amount of detail. However, you don't need to know all of the aspects of this diagram, and we will talk about which things are not important to know. But I like to show this because we have our NADPH produced, as we said, in the oxidative phase. And we can see what is the function of NADPH. So here's our glutathione. We'll go into detail on this a little bit. Here's our precursors for fatty acids and sterols. So I like this diagram because it actually connects. And these aren't like one NADPH is different from another NADPH. They're just using this they're using the two NADH produced by pentose phosphate pathway to elaborate on what are the two functions. So there's not like step 1 is for glutathione reduction and step 3 is for fatty acid synthesis. There's no difference between these NADPH. They're happening in the same compartment. Then we show our non-oxidative phase. And the other reason I really like this diagram is because it shows how the connections to glycolysis are made. So as we'll talk about, if we, for instance, don't need to use ribose 5-phosphate to make nucleotides coenzymes, then we would actually just send our pentose phosphate pathway intermediates back to glycolysis so that we can harvest ATP from them. So we talked about, we said NADPH is just NADH with an extra phosphate. So here's our NADH structure, and here's our NADPH structure. And we'll look again when we go to the electron transport chain, we'll look again at NADH structure. I just wanted to emphasize here, the extra phosphate is really the only difference between these two. Now, NADH is used for the electron transport chain and for certain reactions. NADPH, on the other hand, is not used for oxidative phosphorylation. The functions of NADPH, as we saw on the previous slide, will be synthesis of fatty acids and steroids, sterols, as well as reactive oxygen species detoxification. So those are the specific purposes of NADPH, and we would make NADPH if we needed to do those specific purposes. And otherwise, we won't make it. Questions on this slide?
How important is it for us to recognize the structure?
Yeah, you do want to know the structures of NADH, FADH2, and coenzyme A, as well as coenzyme Q. So we'll see all of those structures. I think coenzyme A will go into next time, but we will see coenzyme Q we actually did see in citric acid cycle, and we'll see coenzyme Q pop up again in electron transport chain. And you do need to be able to recognize, because an MCAT is not a drawing test, but you do need to recognize NADH, FADH2, and those other guys as coenzymes.
Is coenzyme Q, is that ubiquinone?
Ubiquinone. Mm-hmm. Yeah.
Cool, thanks, Charles.
Sure. So step 1 of pentose phosphate pathway oxidative phase is going to be conducted by glucose 6-phosphate dehydrogenase. So based on the name, what does this enzyme do? Oxidation. Mm-hmm. So it's going to oxidize its substrate, glucose 6-phosphate, and then it's going to reduce what? Close, NADP plus. So similarly to NAD plus, NADH, we have a NADPH is the reduced form. NADP plus is going to be the oxidized. So here's the reaction that this catalyzes. And so here's our glucose 6-phosphate. As we talked about, this can go many places in metabolism. When pentose phosphate pathway is active and glucose 6-phosphate dehydrogenase is upregulated, then we will oxidize our glucose 6-phosphate into what's called 6-phosphoglucanolactone. Don't really worry about the sigma here, delta, sigma, whatever the Greek letter is. Don't worry about it. What is the functional group what is the functional group of the carbon here in glucose 6-phosphate? What is that functional group called? So this guy right here. So this would be an ester, right? So this would be not an ester, but
Is that a hemiacetal?
that is a hemiacetal. So a hemiacetal is when you have a carbon that has both OR connected and OH connected. So we know hemiacetals, do they lead to a sugar being a reducing sugar or a non-reducing sugar?
Reducing.
Reducing sugar. So glucose 6-phosphate is, of course, a reducing sugar when it has a hemiacetal. When it gets oxidized to, so what do we call an ester that's part of a ring? Cyclic ester is called a, hint is in it's in the name.
Lactone?
A lactone. A cyclic ester is a lactone. So it's kind of redundant to have two names for it, whether it's in a cycle or not, but that's the case. So we do see that carbon had 2 bonds to oxygen, and this is called the anomeric carbon, right? This is carbon 1, 2, 3, 4, 5, 6. And now it'll have 1, 2, 3 bonds to oxygen. So we do have an oxidation. So when we oxidize the anomeric carbon of glucose 6-phosphate, it becomes no longer a reducing sugar, first of all. And second of all, we will also use the electrons that we gain from oxidizing that bond to reduce NADP plus into NADPH. So we'll lose a hydrogen from here on carbon, the anomeric carbon, and we'll lose a hydrogen from here on the hydroxyl. So one of those hydrogens will be a hydride, which is an H with 2 electrons and a minus, hydride H minus. We'll go to NADP plus, and that's why we lose the plus charge. This is similar to NADH. And we will also have the other hydrogen, I don't remember which one it is, I think it's this one. This other hydrogen will also be lost as H plus into solution. This enzyme, glucose 6-phosphate dehydrogenase, is inhibited, of course, by NADPH. That would make sense. If we have NADPH, and this is the committed or rate limiting step of pentose phosphate, if we have NADPH, it would make sense that we don't want this enzyme to be active. Don't worry about G6P deficiency. It's also inhibited by fatty acyl-CoA. So why might that be? Why might this enzyme be inhibited by fatty acyl-CoA? It's a tough question because we haven't gone over fats yet.
Is it used in fatty synthesis? Is that why?
Yes, 100%. Yep. So because NADPH, as we said, is used in fatty acid synthesis, if we have a fatty acid, which is in the form of a fatty acyl-CoA, that's a fatty acid that's on its way to beta oxidation. So if we therefore have that, we don't need to make more fatty acids. So therefore, it would make sense that we would inhibit an enzyme that makes NADPH, which is used for fatty acid synthesis. Excellent. This is an irreversible step. So we said functionally, why is it an irreversible step? Well, if we're doing the step, we need NADPH, we don't want to go backwards. Chemically, why this step is irreversible? So that can't be the whole story. We have seen reversible reactions with NADH and NAD+, which we talked about when we mentioned that we're going to go back to the malate shuttle again today. When we talked about the malate shuttle in gluconeogenesis, remember the problem was that oxaloacetate is an intermediate between pyruvate and phosphoenolpyruvate PEP. Is an intermediate between those two in gluconeogenesis, but oxaloacetate doesn't have a transporter. So what happened was oxaloacetate got reduced to malate using an NADH, and then malate has a transporter, a shuttle, which we'll go into again today. Malate is then in the cytosol. Malate can then be oxidized back to oxaloacetate. So that's when and then we will get back actually our NADH that we used to reduce oxaloacetate to malate in the matrix of the mitochondria. So that is a reversible reaction, and we get our NADH back that we put in. Likewise, we said lactate dehydrogenase is another example of an enzyme that leads to either production of NAD plus from NADH or vice versa, production of NADH from NAD plus. It's another reversible reaction. Remember that One of the functions of lactate, of course, is in fermentation. In the Cori cycle, when we send lactate over to the liver will then make lactate back into pyruvate and do gluconeogenesis on pyruvate and send glucose back to the muscle. So it's Cori cycle. But also, one of the things we mentioned with gluconeogenesis is with lactic acid fermentation is that lactate is also sort of a reserve storage of pyruvate for when the citric acid cycle is backed up and we don't want to send pyruvate into the mitochondria until the citric acid cycle has used up more of its substrates and has room to take on more substrates. So both of these are reversible reactions. So why would this one be irreversible? We also mentioned hexokinase, phosphofructokinase, and pyruvate kinase from glycolysis were considered irreversible because of the large negative delta G naught. And in this case, the reason why this is irreversible is that in practice, the lactone product here is removed too rapidly. It's processed by the next enzyme in the pentose phosphate oxidative phase. It's processed so quickly that it actually just doesn't have time to go back. And as well, we said functionally, there's no reason why we'd ever want to make NADPH back into NADP plus via pentose phosphate pathway, because that would defeat the whole purpose. So functionally, this is why. Chemically, this is why. Any questions here? So remember, one of our themes from glycolysis in the citric acid cycle was that when a carbon to oxygen bond got oxidized, and NAD plus got reduced to NADPH. And we have a variation on that theme right here. Carbon to oxygen bond got oxidized, and an NADP plus got reduced to NADPH. So this is similar to our themes from before. This is one of the things that I mentioned when it comes to looking for patterns. We can connect this pattern here that happens in the pentose phosphate pathway to something that seems totally different in NADH and NAD plus, really just the same thing happening with a phosphate on NAD. Step 2. So we've now made 6-phosphogluconolactone. We have an enzyme called 6-phosphogluconolactonase. It's a fun one. What do we think this enzyme might do?
Remove the lactone.
Yeah, so it's just all we can tell based on this name is it sounds like it's doing something to the lactone or to the 6-phosphogluconolactone, hydrolyzing something, breaking down something. So what do we see happen? We have our 6-phosphogluconolactone in the ester form. We have a water come in, and the 6-phosphogluconolactonase will then break apart that ester. We go back to the linear form of glucose. So this we could think of as an acid catalyzed ester hydrolysis, which would make this enzyme what class?
Lyase?
It's a good guess. We have used lyase to break open a lot of things. We had aldolase, we had fumarase.
Hydrolysis.
Hydrolase, yep. So this will be a hydrolase. And this should make sense because how do we make an ester? Like, what are the two reactants in a Fischer esterification, for instance?
It's alcohol and carboxylic acid, maybe. That one.
Yeah, the two reactants of a Fischer esterification are an alcohol and a carboxylic acid. So if we're hydrolyzing this ester, we should be able to find a carboxylic acid and an alcohol. So we have carbon 1, which should be right here. And here's our carboxylic acid. 1, 2, 3, 4, 5. Carbon 5 should now have an OH. So if you look 1, 2, 3, 4, 5, carbon 5 is back to its OH. Now, let me ask you this. If I were to radiolabel the oxygen right here, would that oxygen end up in the hydroxyl, in the carboxylic acid OH, or would it end up in the alcohol OH?
I think alcohol OH. That one is closer to the phosphate, but I don't know.
Yeah, you would be correct. Yep, the alcohol and the penultimate carbon. Good, good, good. So if we think sort of about mechanistically what's happening here, we have a water is attacking a carbonyl carbon, breaking open the carbonyl, and we would end up with a tetrahedral intermediate that would then have the oxygen come back, restore its pi bond with carbon, and break off the oxygen of carbon 5 as the leaving group. And so our radio label would end up on carbon 5's oxygen. Great job.
But would it be okay to just say, okay, that oxygen is closer to phosphate than, but it's not really closer, it's in equal distance, right, from the alcohol. So like, is there an easier way to tell where that radio-labeled oxygen will end up if I don't know what will happen once it's hydrolyzed and where the bonds go, etc., etc.?
I would say not necessarily, only because, well, so we know sort of our like carboxylic acid derivative reactions that they all kind of take this form where a nucleophile attacks the carbonyl of the carboxylic acid derivative, breaks the carbonyl bond, becomes like an O minus, and then kicks off the leaving group. So all carboxylic acid derivative reactions occur via a similar mechanism. So that's what I would say to incorporate your knowledge of OChem reactions, because the position of the oxygen here is won't necessarily lead us to the conclusion that the radio label ends up here, because we don't know, is this the bond that's being cleaved, or is this the bond being cleaved, unless we know our mechanisms. Yeah, so I hope that helps.
Thank you.
Yeah, I think eventually I'm going to have a series of OChem reaction videos on YouTube, but I haven't started working on that project yet.
Yes, please.
Yeah, I literally have like a lecture that I used to deliver when I worked for a company, which I'm not going to mention here. I have a series of reactions that are the main high yield reactions on the MCAT. So I already have the notes. I just need to put it up. I just need to record the lecture. So that could be something that we do in future sessions together. One more thing, go ahead. Oh, one more thing to point out, we can connect the name here as a gluconate to, would this carboxylic acid be protonated or deprotonated at physiological pH? Protonated or deprotonated?
Deprotonated?
It'd be deprotonated. So a deprotonated carboxylic acid has what name? Carboxylate. So that's why the name ends with 8. So mainly just recognizing the names here. I don't think the structures are super important for you to memorize. So recognition of these names. If you see a gluconolactone, you know you're in the pentose phosphate pathway. If you see a gluconolactonase, you definitely know you're in the pentose phosphate pathway. And if you see a gluconate, you're definitely in the pentose phosphate pathway. What I like to say is if you ever see like a weird sounding sugar that you don't remember from any other metabolic reaction or pathway, that's a pentose phosphate pathway intermediate. We said this was a hydrolase, used water to break ester into carboxylic acid and the 5' hydroxyl. That's all I have for that slide. Any questions? Next is 6-phosphogluconate dehydrogenase. So we can predict that this enzyme is going to do what to the substrate? It's going to oxidize its substrate and reduce, yes, NADP plus to NADPH. So here's what we have here. We have our 6-phosphogluconate, our dehydrogenase. Whoops, I cut off part of the NADPH, but you know it's here. And then what happened to the number of carbons?
Oh, I see. So it's becoming a ribose.
It's becoming something similar to ribose here. So what carbon would this be if we use the original numbering? What carbon would this guy be? Be 1 or 2? This would be 2. What happened to carbon 1?
If it's oxidative, maybe just CO2, but like.
It does, it becomes CO2. So we know that carboxylic acids can be decarboxylated. So that indeed is what happens here. We lose carbon 1 as CO2. The only deficit of this figure is they didn't show the CO2. I would have preferred that, but here we go. Here's our CO2, carbon 1. We have carbon 2, 3, 4, 5, 6. So in our carbons 2 through 6, did any carbon get oxidized? Carbon 3 got oxidized, right? So carbon 3 was a hydroxyl, became a ketone. So that is where our electrons are coming from. Here's our H, and we'll end up with NADPH and H+. Questions on the sort of what's going on here? Okay, I will.
A question.
Yes.
So, for the mechanism, it would be decarboxylation for the CO2 to come off, but what would there be some sort of mechanism we should know for the hydroxy becoming the carbonyl?
Mainly that the carbon and the oxygen both lose a hydrogen and gain a bond to each other. And that the two hydrogens that are lost, also one of them has 2 electrons as a hydride. And the hydride is what adds to the NADP+.
Okay. And are any changes at chiral centers something we need to take note of when we're doing our studying, or is this not, that detail is not required?
It's good. In some cases, it will be important. For instance, I mean, like comparing something like glucose versus galactose, which I know we've talked about before. I don't think there's going to be any changes to chiral centers that are important for pentose phosphate pathway. Yeah.
Okay, thank you.
Yeah. There was one that we noted in video 1, which was on citric acid cycle. There was one that we noted there, which was citrate goes to isocitrate, where the hydroxyl goes to carbon 5 of isocitrate. And we talked about sort of some stereochemical changes there. We went from an achiral citrate to a chiral isocitrate, but I mean, other than that, I haven't really found it necessary or important. Now we have ribulose 5-phosphate, which is going to be similar to ribose 5-phosphate. What's the difference? Ribose versus ribulose. Do we know what ribose looks like? Maybe they got a good mnemonic.
Does it have to do with like it being aliphatic and cyclic?
That's a good guess. In this case, it's not so much that we care about that. It is so Nikki says in the chat, ribose is an aldose, ribulose is a ketose. Excellent. This is for y'all, is this my right hand? It's my right hand, right?
Yeah.
Okay, cool. This is my left hand in real life, but yeah, I know that the screen is flipped. So the mnemonic for ribose is all right. So all of your fingers will curl to the right, and then the top will be the aldehyde. Your thumb will be the aldehyde. So we have carbon 1, 2, 3, 4. Carbon 5 is achiral, but that's the mnemonic. Ribose is all right. And so ribose is an aldose. Ribulose is a ketose. Cool, cool. This is the second irreversible step. What do we think makes this step so irreversible? So for the first step, the glucose-6-phosphate dehydrogenase, we said that was irreversible because the lactone product gets processed by 6-phosphogluconolactonase so quickly that it doesn't have time to go backwards. So in this case, it's a slightly different sort of reasoning as to why this is irreversible. And I guess to prompt your answer, I would say, what thermodynamic property would lead to this being so irreversible? Would it be Gibbs free energy, entropy, or enthalpy?
Enthalpy, entropy, maybe?
Entropy.
Yeah, because they are creating CO2, water, and ribulose, so a lot of things.
Exactly, yeah. So whenever we make a gas during a reaction, the formation of a gas leads to a large increase in entropy, and that makes it hard for the reaction to go backwards. Good. So the decarboxylation is what makes this so irreversible. We also have our first pentose in the pentose phosphate pathway with ribulose. Of course, it'll be a keto pentose, whereas ribose will be an aldopentose. Again, mainly just recognize the names here and recognize that this is the second NADPH producing step, and that would make sense if you can connect it to a dehydrogenase. Any questions? Oh, yeah. This is the last step of the oxidative phase. So oxidative phase overview. We started with glucose 6-phosphate, which instead of going into glycolysis, went into pentose phosphate pathway. We oxidized glucose 6-phosphate to 6-phosphoglucanolactone using glucose 6-phosphate dehydrogenase. We reduced NADP plus to NADPH. Now we're going to talk about this GSHG, GSH in a moment. And then we did a hydrolysis, and then we did another oxidation, making 2 equivalents of NADPH, which we can use for the stated functions. The oxidative phase is irreversible. And during the oxidative phase, we changed our substrate from 6 carbons to 5 carbons. This phase is active when NADPH is low. Any questions before we move on to the non-oxidative phase? Or actually, I think we're going to talk about glutathione first. Okay, so NADPH and glutathione. So NADPH, being a reduced electron carrier, can then go ahead and reduce other guys. In this case, we can reduce glutathione disulfide, which is this guy, into glutathione, which is this guy. What types of precursors is this glutathione made from? What types of precursors? Some carboxylic acid styles and amide.
Is that a cysteine and a glutamine?
So we have a cysteine. This would be what amino acid on the end here?
Serine?
So this is the alpha carbon. So the side chain is just a hydrogen.
Oh, yeah.
I see.
Glycine, good. And then it's a little hard to identify, but this amino group in the process of making glutathione, this amino group used to be here. And it was isomerized to over here. So if we pretend the amino group is here, this is the alpha carbon, the side chain is 1, 2, 3 carboxylic acid. What amino acid was that originally?
Glutamate.
Glutamate, glutamic acid, excellent. Yeah. So that is what glutathione is made from. And we utilize the cysteine sort of residue to perform redox reactions similar to disulfides in protein structure, right? Once we have reduced glutathione disulfide, GSSG, to glutathione, GSH, glutathione can then reduce reactive oxygen species such as peroxide into water. So that is the benefit, as one benefit of making NADPH, is it can go restore reducing power to our cellular antioxidant, glutathione. And then here's the version that we saw in the other figure, where we reduced NADPH to, NADP plus to NADPH, then NADPH reduces glutathione disulfide to glutathione, and then glutathione reduces peroxide to water. So any questions here? Cool. Now, non-oxidative phase. We'll start with ribulose 5-phosphate, our final product of the oxidative phase. We can go off in a number of different directions from there. You can see this is called transketolase, not important in my opinion. This is transaldolase, again, not important in my opinion. But these will be doing aldo and retroaldol reactions. And then ribose 5-phosphate, of course, we said can be used to make nucleotides coenzymes. And purpose, therefore, of the non-oxidative phase would be, and we'll look at this on the next slide, returning intermediates to glycolysis post NADPH production. So for instance, if we need NADPH, but we don't need nucleotide precursors, such as from ribose 5-phosphate, then we don't need to go into the ribose 5-phosphate and send that over to organelles that are going to make nucleotides. Instead, we can have our intermediates go back to glycolysis, so then we can harvest ATP from them. Or if we wanted to make glucose out of them, we could go do gluconeogenesis potentially as well. But largely, when we send these intermediates, and we'll look at this on the next slide, we send them back to glycolysis largely is to make ATP from them. And then if we do need to make ribose 5-phosphate, or for nucleotides or coenzymes such as NAD, FAD, or CoA, then what phase of the cell cycle would the pentose phosphate pathway non-oxidative phase be most active in?
Mitosis.
Close, but we actually, before mitosis, we have to do what? We have to do DNA
Synthesis.
synthesis. Yeah, S phase, perfect. So S phase would be when this is most active, or maybe it would start in G1. I'm not 100% clear on that. But in S phase, we know we need to replicate all of our DNA, make an entirely new set of DNA, and that's going to use lots and lots and lots and lots and lots of nucleotides. So we'd expect the non-oxidative phase of the pentose phosphate pathway to be highly active during S phase, or at least prior to S phase and prior to DNA replication. In my opinion, for the non-oxidative phase, don't memorize the names necessarily, just be able to connect what I call weird sounding sugar names to the pentose phosphate pathway. If you see something like xylulose, pseudoheptulose, erythrose, if you see something like that, where you're like, I'm pretty sure that's not in any other metabolic process that I remember, it's a pentose phosphate pathway intermediate. Any questions here?
Yeah, you said that the beginning molecule is the you said it's the ribulose, right?
Yeah.
All right, cool. Thank you.
Yeah. Person in the chat, Kiana says, sorry, can you go over why the GSH can reduce the reactive oxygen species on the last slide? Yeah. So this is a thiol functional group. This is what cysteine looks like when it's not in a disulfide. And we can lose, and imagine there's two equivalents here. So there's two glutathiones. We can take 2 hydrogens and 2 electrons, and we can send them to a peroxide, for instance. And so we can turn the oxidation state of oxygen in peroxide, which is what? What is the oxidation state of oxygen in a peroxide? That's one of the exceptions for oxygen.
-1.
-1. So normally oxygen is -2. So we can see we're going from -1 to -2 in water, and therefore oxygen's gaining electrons. And we're also going from 2 hydrogens to 4 hydrogens. So we're going to take our 2 H's from the 2 thiols, as well as 2 electrons, take away 2 H's, 2 electrons, link up our sulfurs, send the 2 H's and the 2 electrons to peroxide, where peroxide can then get reduced. Does that answer your question? Awesome. And then the pentose phosphate pathway, as we alluded to on the previous slide, also has connections with glycolysis. So for instance, if we needed to make NADPH, remember the top is irreversible, the bottom is reversible. If we needed to make all of these reactions down here are reversible reactions. So for instance, xylulose 5-phosphate and ribose 5-phosphate could be used to make glyceraldehyde 3-phosphate and pseudoheptulose 5-phosphate. So those are like the 2 inputs and then the 2 outputs. Likewise, G3P, GAP or whatever, and pseudoheptulose 7-phosphate, those guys can get together and make erythrose 4-phosphate and fructose 6-phosphate. So all these reactions have very close to 0 delta G naughts, so they're very easy to reverse. And we could see that our glyceraldehyde 3-phosphate could go into glycolysis. We could say our fructose 6-phosphate could go into glycolysis, or we could send a xylulose and an erythrose and then make both using actually the same enzyme here, and make both fructose 6-phosphate and glyceraldehyde 3-phosphate. So if we don't need to use ribose 5-phosphate to make nucleotides or coenzymes, we can then just go back to glycolysis, as we've been alluding to. In my opinion, just know these connections exist. I don't think you need to know exactly where the pentose phosphate pathway glycolysis connections are for the MCAT. remember the pentose phosphate pathway occurs in the cytosol as well as glycolysis. So these connections can be easily made with no intercompartmental transport. Questions here?
Can we assume that then prokaryotes also undergo pentose phosphate pathway, just like glycolysis?
Most definitely. Yep. They actually invented it. We inherited it as eukaryotes.
Okay, thank you.
You merely adopted the pentose phosphate pathway. I was born in it. Okay, Batman. Yeah. Oh, here's our overview.
Brilliant reference, by the way.
Now, onto the electron transport chain. So we've been making all this NADH from glycolysis in the citric acid cycle and the pyruvate dehydrogenase complex. We're making FADH2 in the citric acid cycle. Now let's talk about how do we get vast amounts of ATP from these electron carriers. Remember, glycolysis was only generating 2 ATP net. Citric acid cycle generated per glucose 2 GTP. We've only got 4 ATP equivalents just based on what we call substrate level phosphorylation. So those ATPs and GTPs that we get directly from glycolysis in the citric acid cycle is what we call substrate level phosphorylation, which is as opposed to oxidative phosphorylation. So the rest of this lecture is going to be about oxidative phosphorylation and how do we get there using the electron transport chain? How do we get vast amounts of ATP from our mitochondria? The electron transport chain utilizes the reduced electron carriers from carbohydrate and, as we'll see later, fat catabolism to produce large amounts of ATP. It is located along the mitochondrial inner membrane. So two terms that are going to come up a lot in the rest of this lecture are inner membrane and intermembrane space. So I'm going to be enunciating my T's very obnoxiously, just so we're clear when I'm talking about inner membrane versus intermembrane space, okay? NADH and FADH2 will be oxidized in the electron transport chain. So remember, these guys were formed from the process of reduction. And finally, they're going to become oxidized and shuttle their electrons through the electron transport chain through proton pumping complexes that we will then use to establish a proton gradient. Ultimately, oxygen is known as the terminal electron acceptor. So at the end of the electron transport chain, as we'll see in complex 4, oxygen will then be reduced to water. So because of this, our electrons, we're sending them through this complex pathway, but they need oxygen at the end in order for the pathway to be completed. So if we were in anaerobic conditions, if we had no oxygen, eventually the electron transport chain would be backed up and we wouldn't be able to make any ATP at the end, or we'd be able to make very little ATP at the end. So the electron transport chain is only active in anaerobic conditions, as we said, and likewise, the citric acid cycle and the pyruvate dehydrogenase complex are only active in aerobic conditions because those guys make most of their energy from electron carriers that need oxygen at the end of the electron transport chain. So what we said, we talked about anaerobic metabolism last week, and we talked about lactic acid fermentation. And so this will only be active in aerobic conditions. These proton pumping complexes will set up a proton gradient, which is an electrochemical gradient. It has an electrical gradient and a chemical gradient. And this gradient is located between the mitochondrial intermembrane space and the mitochondrial matrix. Finally, ATP synthase will couple the proton gradient with ADP phosphorylation. We've talked a lot about coupling when it comes to coupling non-favorable reactions with ATP hydrolysis. We're doing the opposite now. We're coupling proton gradient to make ATP. So that's what we mean when we say coupling. It is unfavorable, non-spontaneous to phosphorylate ATP from ADP. It's unfavorable. So we're turning it favorable by coupling it with this proton gradient that we set up in the electron transport chain. So that's our overview of the electron transport chain. Any questions before we go into deets?
Yeah, quick question. You said that the proton gradient is an electrical gradient or electrical chemical gradient?
It's going to be both electrical and chemical. And we'll show exactly why that is.
Cool.
Mm-hmm. And what couple words we use when we talk about this ADP phosphorylation at the end of the electron transport chain are oxidative phosphorylation, sometimes abbreviated as Oxphos, it's fun to say, And there's a word that you may see come up. It's actually in one of the AMC full lengths, if I'm correct, which is chemiosmosis. So chemiosmosis is another name for oxidative phosphorylation. So if you see chemiosmosis, they're talking about oxidative phosphorylation. They're talking about making ATP from the electron transport chain. So we'll start our journey into electron transport chain with endosymbiotic theory, which is the origin of the mitochondria. When I wrote this slide, I felt like it sounded like like a Marvel movie like title, mitochondrial origins. So mitochondria were originally a primitive a bacteria. Think of it as a Pac-Man. Yeah, we think about these mitochondria. How do we get them? Well, mitochondria was a bacteria that we adopted as a pet long ago, back when eukaryotes could only do anaerobic metabolism. So we had this bacteria that had evolved to, and I believe it's, nope, I don't know, I'm not going to say what kind of bacteria it is because I don't remember. This bacteria evolved to do aerobic respiration before we did. So we made a deal, we made a bargain. If you come live in our cell, we will give you food and water, we will feed you with molecules, and what you will give us is you will give us lots and lots of ATP. So what the benefit we're providing in this symbiotic relationship to the bacteria is we're making sure the bacteria, our pet, never goes hungry again, always has access to food. In exchange, the pet mitochondria gives us tons and tons of ATP. A pretty good deal from both perspectives. And so I like to show this slide because it is the reason for the mitochondrial double membrane. There's the membrane of the bacteria itself, which is what we'll call the mitochondrial inner membrane. And then there's the outer membrane, which is actually eukaryotic in origin. So this is the outer membrane, this is the outer membrane, and this is why they have a double membrane. One that they originally had, the inner membrane, and one that they got when they call it invaginated into our cell, the outer membrane. I think of outer membrane is like vesicle. Yeah, exactly. It's like a vesicle. Yeah. So based on this, where do bacteria perform their electron transport chain? Do they have their own mitochondria?
Membrane.
Yeah. Bacteria perform the electron transport chain on their plasma membrane. So their complexes, which are similar to ours because we got it from them, we have our mitochondria from them, their complexes are actually on the plasma membrane. And they will actually pump protons where? Where do they establish the proton gradient? Outside. Yeah, they actually, it's super weird to think about it that way, but they actually will pump protons into the extracellular fluid. Or I guess if you're a gram-negative bacteria where you have an inner membrane encased by a cell wall coated by another outer membrane, if you're a gram-negative bacteria, I guess it would be between your plasma membrane and your outer membrane. So yeah, they'll actually pump the protons outside of their plasma membrane. Any questions here?
I do actually. Do we, for the MCAT, need to know the difference between gram positive and negative?
I'd say it'd be good to know. Yeah.
Okay.
Of course, so gram positive, they just have the one membrane coated encased by a cell wall. Gram negative, the plasma membrane, cell wall, and then outer membrane. Do you need to know details of gram staining? Probably not details of gram staining, but if you ever if you can connect the details of gram staining as to why bacteria are gram positive or gram negative, that could only help, I believe.
So negative has essentially two membranes plus a cell wall.
Yeah, let's just, why not? Let's do it.
Oh, okay. My bad, guys.
Oh, no. This is good for everybody. So here's sort of a simplified diagram. So a gram-positive will have the plasma membrane. And so we define plasma membrane as the membrane that encases the cytosol. So they have their plasma membrane, they have a little bit of space, and they have peptidoglycan. Peptidoglycan is fairly porous, so stuff can get in and out of peptidoglycan. What is the main purpose of having a cell wall? For protection against?
Exterior, outside.
I was gonna say for things that engulf or break down the bacteria. It's like a defense mechanism.
Mm-hmm. And the most common way for a bacteria to actually be destroyed by external conditions, the most common would be osmosis. So it is protection against osmolysis, because if you were hypotonic, if the outside solution has a lower ionic concentration than inside your cell, where's water going to want to flow?
Oh, okay. I see where you're going with this.
Inside the cell.
Inside the cell. I saw a cool mnemonic the other day. It was hypotonic. Capitalize the O in hypo and think of it as a swelling cell, because in hypotonic conditions, your intracellular osmolarity is higher than the outside environment. So water wants to flow in to normalize the ionic concentration between the inside and the outside of the cell. So that's mainly the reason why these bacteria have peptidoglycan, is for protection against osmolysis. So then details of the cell wall for gram-negatives. So here's the, it's a lipopolysaccharide. Not lipopolysaccharide, peptidoglycan. Lipopolysaccharide is part of it. And that's lipopolysaccharide, like a super antigen. We'll learn a lot about this in med school. Or if you've taken microbiology, you probably already did learn a lot about it. And then with gram-negatives, this is just the cell wall, actually. I don't want to look at that. With gram-negatives, they have the plasma membrane, then they have the cell wall, and then they have the second membrane outside. So when it comes to gram staining,
Is the peptidoglycan the cell wall?
yes. Yeah.
Okay.
When it comes to gram staining, a 4-step process. You first add what's called crystal violet, and everybody gets this crystal violet and looks purple. Then you add iodine. What happens is crystal violet and iodine are buds. They're going to link up and they're going to form this complex that gets stuck in a cell wall. So if you're a gram-positive bacteria where your cell wall is exposed to the external environment, if you're gram-positive, that crystal violet and iodine complex can get stuck in your cell wall. And then if you're gram-negative, they actually can't get in because they can't go through the outer membrane. So then we'll then add alcohol to wash everything away. But in the gram-positive, because the crystal violet iodine complex gets stuck in the cell wall because it's exposed to the external environment, the alcohol won't be able to wash that away. With gram-negative, since it was just like, it was just there, it wasn't like in their cell wall or anything, it was just outside their membrane, they will get washed away and they will become colorless. So we got our purple gram-positives who have the crystal violet iodine complex stuck in their cell wall. We've got our clear gram-negatives who have the slippery outer membrane and everything gets washed away. The last step is adding a counterstain called safranin. And the safranin will coat everybody, but the purple like sticks out more for gram-positives. So they still look purple. But then now the gram-negatives will look pink. So that's gram staining, and that's why we have difference between gram-positive and gram-negative. So why is this important for medicine? Which one do we think would be harder to treat? Infections by gram-positives or gram-negatives? Gram-negatives, right? So they have a slippery outer membrane, right? Where it's harder to get stuff in there, antibiotics. It's harder to get antibiotics to actually like break down the cell and cure the infection. So yeah, so you're gonna have different antibiotics for gram-positive and gram-negatives. Yeah. Any other questions while we're on that topic? Cool. All right, let's talk a little bit more about mitochondrial structure. So here we have our outer membranes. This is inside of us now. The outer membrane is porous to small molecules. Proteins, when we get to the level of size of like a protein, proteins can't go through these pores. But for all intents and purposes, metabolic molecules or electron carriers like NADH, pyruvate, have no problem getting through the outer membrane. On the other hand, the inner membrane, right, which is where we're going to set up the proton gradient, the inner membrane is impermeable to most things other than short chain and medium chain fatty acids. So the inner membrane is a lot trickier to get through. And so a lot of molecules actually have transporters located on the inner membrane where they can just diffuse through the pores of the outer membrane, but they have special ways of getting into the inner membrane. Such as with pyruvate, we call it pyruvate translocase. There's also a lot of folds in the inner membrane. Why would there be these cristae or folds? Why would these cristae be existing on the inner membrane? What are they for? What function do that serve?
Surface area.
That will increase surface area so that we could have extra surface area to conduct electron transport chain and oxidative phosphorylation. Mitochondria will also have its own DNA. What would be the shape of the mitochondrial DNA? It would be linear.
Like bacterial, circular.
Circular like bacteria. Questions here? Don't worry about granules. Don't worry about, that's pretty much the only thing that's like, we're not going to talk about here is the granules. They do have their own ribosomes. So they will make some of their own proteins. Oh, there it is, yeah. Other functions of the mitochondria. So in adipose tissue, we'll have an example on this later after we talk about ATP synthase. In adipose tissue, the mitochondria serve to generate heat during cold temperatures to protect vital organs. This is interesting. I don't think you need to know this for the MCAT, but this was something that I saw that I thought was like, wow, that was counterintuitive. In the gonads and the adrenal glands, the mitochondria is the location of steroid hormone synthesis. I haven't really gone into detail like researching why that is, but just thought that was interesting. So we're just talking about other functions of the mitochondria. And then in all cells, there's the mitochondrial apoptosis pathway. So there's two pathways to apoptosis. There's the intrinsic, which is the mitochondrial, and the extrinsic. So in response to some apoptotic stimulus, such as DNA damage, endoplasmic reticulum stress, oxidative stress from such as ROSs, reactive oxygen species, or viral infection. So 4 different types of stimuli that can lead to activation of the mitochondrial apoptosis pathway. What happens is the mitochondrial membrane loses integrity, and then cytochrome C, which is we'll talk about as a mobile carrier of electron transport chain, cytochrome C leaks out into the cytosol. We would normally never see cytochrome C in the cytoplasm. It should be restricted only to the mitochondria. So if it leaks out, that is an indicator of the mitochondrial pathway of apoptosis. And then cytochrome C will go through, don't worry about this, we'll activate caspases. And caspases, you definitely want to be familiar with those guys in terms of being involved in apoptosis. In the MCAT, we don't really need to worry about it beyond that. So if you have this sort of pathway memorized or learned for the MCAT, then you should be good. There's also the extrinsic pathway, which is typically when you have either like a cancer cell or a virus infected cell. They're not showing it here, but you would present some antigen from either like a cancer antigen or from a viral antigen. Present some antigen, and then a cytotoxic T lymphocyte, a CD8 positive T cell, will come up and be like, yo, we got a problem. This dude is cancerous, or this dude is infected by a virus. We're gonna shut the whole thing down. We're gonna ixnay on the LSA. And then we're gonna and so it gives what they call a death receptor called FAS. It gives it a ligand called FAS ligand, which triggers death domains. And then those trigger caspase 8. And then that will lead to apoptosis. Don't need to memorize numbers for caspases for the MCAT, just be familiar with the caspases and know that there are 2 pathways, the intrinsic and the extrinsic pathway. Any questions here?
Yes, I don't know if you recall, but we actually discussed this in the past. It was ubiquinone that would cause proteolysis or something.
Ubiquinone or
Sorry, go ahead.
ubiquinone or does anybody know which one causes proteolysis?
No, or I confused.
Ubiquitin. Yeah.
Ubiquitin.
So we want to differentiate between ubiquinone and ubiquinol, which are oxidized and reduced forms
Right.
of the of coenzyme Q, and ubiquitin, which you add ubiquitin to proteins that you don't need right now, so you send them over to the proteasome. So yeah, what was your question?
Oh, so I just wanted to be sure because when I was reading how it's involved in the apoptosis pathway, I was trying to recall of something that works in destroying other things, and I wanted to make sure I could differentiate those two and I'm not mistaken them. So thank you.
Yeah, awesome. Yeah, absolutely. Yeah, always good to make these kind of connections. There's also necrosis, which we're not going to really go into here, but necrosis is when, like, I don't know if I can even really describe it. I haven't really prepped necrosis. Necrosis is opposed to, yeah, TNF alpha is involved in necrosis, I believe. Yeah. Necrosis is bad. It's like an unplanned cell death, can lead to inflammation. Yeah, which is TNF alpha. Apoptosis is programmed cell death. So it's like the cell has a contingency plan. If things go haywire and I get infected by a virus or I start to have DNA damage, which could lead to cancer, I'm going to commit apoptosis and I'm going to like bleb everything together into little like granules that can then be picked up by things like macrophages and recycled for other healthier cells. Mitochondria, we said, has its own DNA. I just like to include this because I think it's cool. We hear a lot about the mitochondrial DNA. Does anybody know how mitochondrial DNA is inherited?
Yes, I know that only, at least in humans and mammals and stuff, only the mother can provide it through her.
Through her egg, yeah, exactly. What was it?
Mitochondrial Eve in the evolution that we all have.
Oh yeah. The common ancestor of all humans is the person we got all of our mitochondria from. Yeah, it is maternally inherited, which we know is because, so sperm, right? Sperm are just carrying, they have an acrosome, which has chemicals for breaking open the protective structure of the eggs. They can inject their DNA. Sperm has flagella for motion, and it has a bunch of mitochondria for propulsion, for ATP synthesis. And sperm has DNA. That's essentially what a sperm is. They don't carry mitochondria. The egg from the mother has a huge cytoplasm. Like the egg to sperm ratio in terms of size is huge. So the egg carries all the mitochondria, which is why mitochondrial DNA is inherited from mom. So I like to include this because we hear about the mitochondrial DNA in terms of genetics, but we don't really think about what's actually on that mitochondrial DNA. So mitochondria encodes a number of gene products that it uses for respiration. Now, it doesn't have a DNA polymerase and an RNA polymerase, so we don't really need to worry about those for the purpose of the MCAT, but if you were curious, its DNA polymerase and RNA polymerase are actually nuclear encoded. So the nucleus will transcribe RNA for the DNA and RNA polymerases, and then we'll translate it into cytoplasm, and we'll send it to the mitochondria so that they can replicate their DNA or they can transcribe their genes. On the mitochondrial DNA, we have encoded transfer RNA. So we said they have ribosomes, so they can synthesize their own proteins, at least some of their own proteins. They're going to need a lot of proteins from their parent, which is the cell that has adopted them. They need a lot of proteins from that too. They will encode tRNAs for carrying amino acids. They will encode their own RNA for ribosomes. It has a different number than ours. Don't worry about the numbers for mitochondrial ribosomes. What are the numbers for our ribosomes? The sedimentation units. Eukaryotic ribosomes are 30, 50, 70 or 40, 60, 80?
40, 60, 80.
40, 60, 80, yeah. So 40 small subunit, 60 large subunit, 80 overall. And then bacteria or prokaryotes are?
30, 50, 70.
30, 50, 70. So I just remember that the even numbers go together and the odd numbers go together. Remember, they're not supposed to add up to the overall number because it's not like a mass, it's a sedimentation unit, it's a density unit. So it's like, where do they end up during centrifugation? The small unit ends up at the 40s level, large ends up at the 60s level, and the overall would end up at the 80s level for us. So I remember that the even numbers go together and the odd numbers 30, 50, 70 go together. And then I remember, this isn't why, but I remember that like eukaryotes are larger than prokaryotes. So we got the 40, 60, 80, which are bigger than the 30, 50, 70.
One way I remembered is that, like, if you think about professional athletes, they're usually at the age of 30 years old. Like, that's their peak age. So prokaryotes, pro athletes, 30, 50, 70. I don't know if that'll help people, but
That's cool. I love hearing everybody's like personal mnemonics. That was mine. Mine is like small, big. You got you.
So just make sure I heard you right, because it's super loud on that right now. Mitochondria, they have their own ribosomes, and they're able to encode certain things like tRNA and rRNAs. But then there are also things that the nucleus makes that helps them out too.
Yeah, exactly. So they don't encode everything. Like, for instance, we can see that complex 2, for instance, is not here. They don't encode everything. They just think it's like a matter of convenience. So like when the bacteria that became our mitochondria entered our cells, they probably carried everything from the bacterial genome. Over time, because our cell can do so much of the same things, over time, a lot of those genes became redundant. And I think even some of their genes actually got transferred to our nucleus. So like, it's like it's not vestigial genes, because they actually are using all these genes. Like, but the ones that remained are all related to mitochondrial function. So we have like our electron transfer chain proteins, our complex 1, 3, and 4. We have some of our ATP synthase subunits, and then that's basically what I wanted to emphasize here. Any questions on mitochondrial genome? All right. And yeah, this slide is just for background reinforcement. You don't even know anything here, really. Just know it's circular and maternally passed. Now we're getting to the nitty gritty of the electron transport chain. One thing I want to emphasize here, there's 2 types of diagrams you might see regarding the electron transport chain. There's a diagram, and particularly when it comes to complex 4. So for now, we're just going to note that our diagram shows one NADH entering the electron transport chain, or one FADH2 would be right here, entering the electron transport chain, results in the reduction of half of an O2 molecule and the pumping of 2 protons. Of course, we know that we're not going to have half of an O2 molecule actually in practice. So we'll see 2 types of diagrams describing complex 4. From the perspective of an electron carrier, complex 4 will reduce half of an O2 molecule and pump 2 protons. You can also see our proton gradient is represented by these pluses. So the proton gradient we said was electrochemical. So there's the electrical potential, which is that there's more H plus on one side and more OH minus on the other side. That's the electrical gradient. We can think of it as a potential. And we also have chemical potential or chemical gradient, which is the H pluses themselves, because there's a lower pH, more H plus on this side, and there's a higher pH, less H plus on this side. So there's sort of 2 components of the proton gradient in terms of electrical and chemical potential. Questions here? All right. So as promised, we're going to look at the electron carriers. We kind of glanced at NADPH, and we saw NADH while we're looking at that. We want to also see the oxidized and reduced forms of these guys. So we have our oxidized form of NAD, which is NAD plus. We'll gain an H minus, and typically, as we saw, when NAD plus gets reduced to NADH, the substrate is losing 2 protons, an H plus and an H minus. The H minus joins NAD plus, makes it into NADH, and then the H plus just goes off into the solution and does H plus. So they show the H plus over here because the H plus doesn't combine with our NAD. What does NAD stand for?
I think it's nucleoadenine dinucleotide, or Yeah, nicotine adenine dinucleotide. I think that's it.
Nice, yeah. So it's nicotinamide, which looks like this. Just because we all know about like nicotine being a chemical, let's also just glance at, it wasn't important enough to include it. We'll glance at what nicotine itself looks like. So nicotine itself is going to look like this. It has sort of like a pyrimidine ring, or sorry, not pyrimidine, pyridine ring, and then sort of like a cyclic nitrogen group over here. So that's nicotine. Nicotinamide has the pyridine ring, but then an amide. So that's where it gets its name from. It's similar, but I don't even think that they're actually like, I don't think nicotinamide is derived from nicotine or anything like that. So I think it's like just a similar structure. And then adenine dinucleotide. So here's our nicotinamide. You see our adenine? Adenine would be right here. And then dinucleotide for the 2 riboses. Yep. What's important about this is you should be able to recognize NAD, and you should know that it's a 2-electron transfer. Questions here? FAD. So FAD, what does that stand for? Flavin, yeah. So if you look at like nutrition facts label, you'll sometimes see like riboflavin, which is a vitamin B. So flavin is this dude up here, this big ring structure, easy to differentiate it from nicotinamide. And then adenine as well. And then dinucleotide. So we can see here's one ribose. Where's the other ribose? It's a dinucleotide. There should be another ribose. Where's that at? It's linear. Yeah, it's in the Fisher projection right here. Yep. That's our flavin adenine dinucleotide structure. And then here's the oxidized and reduced forms of FAD. Unlike NAD+, which only gains an H- and releases an H+, FAD will gain 2 H+ and 2 electrons. So we'll go from this structure, and we'll reduce it to this structure. So that's why our FAD can actually gain 2, and that's why we call it FADH2. And then it doesn't have any charges. There's no gain or loss of charge like there is with NAD+, because it's gaining 2 pluses and it's gaining 2 electrons, so it doesn't change charge. If you're ever curious why FAD doesn't change charge, but NAD+ does change charge, this would be why. The electron transport chain doesn't only give away 1 electron. We will get to that. Questions here? It has to do with cytochrome C, is the long story short. All right, so don't get overwhelmed. Nope, nobody freak out here on me. Here's the malate shuttle. I'm gonna have to move my camera to a non-important part of the slide. So this is called the malate shuttle. Remember, we encountered the malate shuttle in gluconeogenesis. So we were describing in gluconeogenesis when we're in the matrix and we want to convert pyruvate back to PEP. Pyruvate would be in the matrix, and then pyruvate would go through the intermediate, which is oxaloacetate. And an oxaloacetate in the cytosol will go to PEP, phosphoenol pyruvate, under action of what enzyme? The enzyme converts oxaloacetate to PEP. It's called PEP carboxykinase. So oxaloacetate, once in the cytosol, would then be converted to PEP via PEP carboxykinase. The problem is oxaloacetate doesn't have a transporter to get through this impermeable intermembrane, or inner membrane, there we go. It doesn't have a transporter to get through the inner membrane. So what happens first is it will become reduced to malate. NADPH will become oxidized. Malate will then go the opposite direction as this figure is showing, because they're trying to show something else here. It'll become malate cytosolic. Cytosolic malate will then get oxidized back to oxaloacetate and reduce NAD+ to NADH. So there's no net gain or loss of NADH molecules. And oxaloacetate can then go to PEP, and PEP can go up the chain of glycolysis, go back up the chain of glycolysis in the process of gluconeogenesis to make glucose. Yeah, so it converts back. So our problem here is that the inner membrane is impermeable to NADH. Remember, we made 2 equivalents of NADH during glycolysis. However, the mitochondrial inner membrane is impermeable to NADH, but we still need to get that NADH from glycolysis into the matrix so we can then put it into the electron transport chain. As we'll see, there's going to be 2 ways of doing this depending on tissue type. So when we want to get NADH into the mitochondrial matrix, we kind of do a sneaky way where we beat around the bush here. We will actually do the opposite of what happens at gluconeogenesis. We'll make NADH into NAD+, we'll oxidize it, and we'll reduce oxaloacetate to malate. So malate, so this is cytosolic oxaloacetate. Malate, and by the way, so remember, the intermembrane space is, for a lot of molecules, continuous with the cytosol because those porins on the outer membrane of the mitochondria can allow conduction of a lot of different small solutes. So we'll see some diagrams which will call this side the matrix and this side the cytosol or cytoplasm. So don't get confused by that either, because remember, the intermembrane space is functionally continuous for a lot of molecules with the cytosol. Once our malate gets transported then into the mitochondrial matrix, we'll oxidize malate to oxaloacetate, which can then go into the citric acid cycle. And then we will get our NADH back. We will reduce NAD+ to NADH. So that's the way that the cell dealt with this problem of glycolysis making NADH, but the inner membrane of the mitochondria being impermeable. Any questions?
So pyruvate can enter the inner membrane space, but it can't get into the matrix without doing the malate shuttle.
In this case, we're talking about NADH. That's also true for pyruvate. Pyruvate also can diffuse into the intermembrane space, and it'll have its own transporter for getting into the mitochondrial matrix.
Okay, so this isn't, so it doesn't have to be compared to. Okay, I totally missed something then. So what is the significance of this? Like,
yeah, let me advance some of these bullet points. But just to reiterate, with pyruvate, pyruvate dehydrogenase complex is located on the mitochondrial inner membrane. So pyruvate will diffuse through the outer membrane into the intermembrane space. And then pyruvate dehydrogenase will spit out acetyl-CoA in the mitochondrial matrix.
Gotcha.
So, yeah.
Okay.
So here what we had is the solution to this problem of the inner membrane being impermeable is that in the intermembrane space, NADH will hand its electrons off to oxaloacetate. Malate will then get transported into the matrix. And in the matrix, then malate will be oxidized back to oxaloacetate, and we'll get our NADH back on the matrix side. Does that make sense? Any questions?
Oh, okay, because we make, via glycolysis, we make NADH outside of the mitochondria. So this is how we get it into the matrix. All right, I'm back, Charlie. I got you.
Yeah. And not important to know for the MCAT, but you probably will encounter in med school that the malate shuttle is expressed in the liver, heart, and kidneys. So the malate shuttle does not sacrifice any energy from NADH. It is a energetically neutral process via that we get NADH from glycolytic cytosol into the matrix, which, as we're going to see in a moment, is as opposed to the glycerol shuttle. So any questions on the malate shuttle while we're here? So to summarize, 2 main purposes of the malate shuttle: to ship oxaloacetate in the form of malate into the cytosol, so then we can do gluconeogenesis, go to PEP, and then et cetera, et cetera. Other purpose of the malate shuttle, to get glycolytic NADH into the mitochondria. So there's another type of transporter called the glycerol shuttle. And so we do glycolysis, we make NADH, and then again, same problem. Inner membrane is impermeable to NADH. Different solution. In the intermembrane space, which again is functionally continuous with the cytosol, NADH can also hand its electrons to dihydroxyacetone phosphate, which we recognize as a glycolytic intermediate. We then are reducing dihydroxyacetone phosphate on carbon 2. into glycerol 3-phosphate. So be careful, this isn't glyceraldehyde 3-phosphate. That would be what happens to dihydroxyacetone phosphate in glycolysis. In the glycerol shuttle, we'll have cytosolic glycerol 3-phosphate dehydrogenase acting in the reverse direction, by the way, and actually not oxidizing glycerol 3-phosphate producing NADH, but reducing dihydroxyacetone phosphate, oxidizing NADH, and then glycerol 3-phosphate being the product. So now we have NADH has handed its electrons off to DHAP and made glycerol 3-phosphate. Now glycerol 3-phosphate's got the electrons, it's got the football, and it's going to hand it over to mitochondrial glycerol 3-phosphate dehydrogenase. Will then reduce FAD to FADH2. So remember, FADH2, or maybe you don't remember because we haven't talked about electron transport chain, at least that part yet. Remember, FADH2 is going to enter the electron transport chain later than NADH. So what will happen here is FAD is just a cofactor for this mitochondrial enzyme. Now, glycerol 3-phosphate dehydrogenase is acting in the way the name suggests. It's oxidizing glycerol 3-phosphate, and it's reducing electron carrier, FAD to FADH2. FADH2 has the electrons now, and it'll give its electrons to coenzyme Q, turning it from ubiquinone to ubiquinol. So this is ubiquinol. Remember, this is similar to succinate dehydrogenase, where succinate gets converted to fumarate, FAD gets reduced to FADH2, and then FADH2 gives the electrons to coenzyme Q. So this bypasses complex 1. Mitochondrial NADH would go into complex 1. So what's happening here is we're actually missing out on protons being pumped by complex 1. And as we'll see, this is going to result in 1 less ATP per NADH. If we take this shuttle, I don't know, it's like a bargain. It's a wish.com shuttle. So we're getting only FADH2 from NADH, essentially. And so we're going to lose out on 1 ATP. And then just one other thing to note is this is also how glycerol catabolism links to glycolysis. So glycerol, we know is a molecule that we can use to make triglycerides if we attach 3 fatty acids, or we can make phospholipids if we attach a polar head and 2 hydrocarbon tails. So glycerol can be catabolized via this process. So it'll become phosphorylated to glycerol 3-phosphate. So that takes an ATP. And glycerol 3-phosphate will then get oxidized to dihydroxyacetone phosphate. We'll send an FADH2 into the electron transport chain. Then dihydroxyacetone phosphate will then continue on with glycolysis. So that's how glycerol catabolism works. I don't know if it's important you know that for the MCAT, but here it is. So questions on the glycerol shuttle?
I just wanted to confirm something quick. So first thing is that just confirming that complex 2 is the only complex that the hydrogen ions aren't going to the intermembrane space at this one, is what you're saying?
Correct. Yep. Okay. So we're not looking at complex 2, but this is analogous to complex 2. Yeah, complex 2 is the only complex of the electron transport chain that is not a proton pump. Yep.
Okay, and the second one is just to confirm. So you were saying how glycerol is a precursor for all these great things like phospholipids and fatty acids.
Triglycerides, yeah.
So what we have here is a glycerol with phosphate, and we catabolize it, and then you have G3P, and then you dehydrogenase.
Correct. Yeah. So glycerol. This would be how glycerol gets catabolized.
Okay.
But it's also a convenient convenient strategy for getting NADH into the electron transport chain from glycolysis.
Okay.
So this is in different tissues than the other one. The other one was heart, liver, and kidneys. The malate shuttle was in heart, liver, and kidneys. So they get the full, like they don't sacrifice any energy when they get their NADH into the mitochondrial matrix. Mm-hmm. This actually sacrifices a little bit of energy because we're not entering the electron transport chain at complex 1.
Okay, so the dihydroxyacetone phosphate's the oxidized form because you remove the you don't have the OH, you have the carbonyl, and then the NAD plus is the reduced.
Yep. The NADH is the reduced.
Right, becoming NADH. sorry.
Becoming, yes,
Yeah.
yeah.
Okay.
So this cycle like has linkages to glycolysis and glycerol catabolism, but the purpose that we're emphasizing here is that it's a solution for getting energy, getting electrons from NADH into the electron transport chain. Finally, let's start talking about the complexes themselves. So we've talked about how does NADH from glycolysis get into the mitochondrial matrix. We talked about the overall process that the goal is to pump protons in the intermembrane space and to oxidize electron carriers, take their electrons and send them into the electron transport chain. So NADH will enter at complex 1, which is conveniently named NADH dehydrogenase. So this is the most common name you'll see. It's an enzyme that oxidizes NADH. So finally, NADH is getting oxidized after being in the process of oxidizing some of the other substrates. It's also known as NADH coenzyme Q oxidoreductase. That's a mouthful. We're oxidizing NADH, we're reducing coenzyme Q, so it's NADH coenzyme Q oxidoreductase. So that is the net reaction here. Coenzyme Q ubiquinone gets reduced to QH2 ubiquinol. We'll look at a structure of those in a moment too. So the first thing that will happen, and then in the process, don't worry about the mechanism because it's still very controversial, the mechanism of how these protons get pumped into the intermembrane space. But the result of one NADH entering complex 1 is 4 protons being pumped. NADH will first hand its electrons to FMN, which is flavin mononucleotide, so similar to FADH2 but different. FMN will then hand off electrons to iron sulfur centers. Don't worry about iron sulfur centers, they're super boring. It's just iron alternating between different oxidation states and being coordinated by sulfur. And then lastly, coenzyme Q will take the electrons over to complex 3. Any questions on NADH dehydrogenase complex 1?
Should we familiarize ourselves with FMN or just focus on the coenzyme and the enzyme itself, or like the name of the complex?
I would worry mostly the name of the complex, linking it to the fact that it's oxidizing NADH, know that it's reducing coenzyme Q. These 2 protons, by the way, are not being pumped, they're just being added to coenzyme Q. I don't know if you need to even know what I'm trying to say, coefficient of protons that gets pumped, but it helps to know that because as we'll see, it'll help us explain why NADH and FADH2 have different ATP yields.
Right.
I don't think you should super worry about FMN, but if you see FMN, just be like, oh yeah, that's in complex 1, I would say.
Ooh.
Yeah, we don't see it anywhere else as far as I'm concerned for the MCAT. Now complex 2, so if you've watched lecture 1 of metabolism, this should look like a familiar figure, and I actually just copied and pasted it because succinate dehydrogenase from citric acid cycle is complex 2 in the electron transport chain. So here's our citric acid cycle. We will oxidize succinate to fumarate, and we will hand electrons to FAD. This is the one step of citric acid cycle that produces FAD, H2. We'll hand the electrons to FAD. FADH2 now has the football. FADH2 hands it over to the iron sulfur clusters. The iron sulfur clusters hand the electrons over to coenzyme Q. Gets reduced to coenzyme Q, reduced form ubiquinol, and then ubiquinol will then migrate over. It's a mobile electron carrier. It'll migrate over to complex 2. This is also known as succinate coenzyme Q reductase. So you can see why that would be the case because we're oxidizing succinate, we're reducing coenzyme Q. It is the, if you get there, it's the only enzyme that participates. Was that? Okay. The only enzyme that participates in both the citric acid cycle and the electron transport chain. As we said, couples the oxidation of succinate to fumarate with the reaction of coenzyme Q, which is ubiquinone going to ubiquinol. FAD is the intermediary. And it's the only complex that doesn't pump protons. So we're not getting any additional proton gradient here. This is just to get the electrons into the chain. Coenzyme Q will then take the electrons over to complex 3. And here's what ubiquinol looks like. Remember, this is different from ubiquitin, which is what you add to a protein you want to target for degradation in the proteasome. This would be our oxidized form of coenzyme Q, ubiquinone, like ketone. Gains 2 electrons and 2 protons. Remember, we saw the 2 protons in complex 1 combining with Q to make QH2. So this is the QH2 ubiquinol. Here are the 2 protons, and it also gained 2 electrons. So it's a 2 electron carrier. Questions here? Yep, so this is all just to get electrons in the electron transport chain. We're not actually making any proton gradient. Awesome. So, okay, let's, again, don't get overwhelmed. This looks confusing, but to be honest, a lot of the complex 3 is like beyond scope of the MCAT. I'm gonna try my best to help explain it. So complex 3 is known as cytochrome C reductase. So we have 2 mobile carriers in the electron transport chain. We have coenzyme Q and we have cytochrome C. Coenzyme Q is to the left or before complex 3. Cytochrome C is after complex 3. And it's also known as Q cytochrome C oxidoreductase. It's oxidizing coenzyme Q and it's reducing cytochrome C. The net reaction of this is we have an input of a coenzyme Q getting oxidized and cytochrome C gets reduced. Cytochrome C, unlike coenzyme Q, only carries 1 electron. So for every coenzyme Q that comes into complex 3, we have to reduce 2 cytochrome Cs to take over to complex 4. The oxidation in cytochrome C is iron 3+ alternating with iron 2+. So that's why it can only carry 1 electron, because it has only those 2 possible oxidation states. And in complex 3, 4 protons will end up in the intermembrane space. So that's a sufficient understanding of complex 3. Now let's see if we can get into the weeds just a little bit without confusing anybody. We'll start with a coenzyme Q reduced that will then enter complex 3, become oxidized, lose protons. So it's a dehydrogenase. We're losing 2 protons, and we're losing 1, 2 electrons. So coenzyme Q got oxidized, lost 2 protons, lost 2 electrons. The first electron will go to a cytochrome C and reduce it to 1 equivalent of cytochrome C. The second electron will actually go to like a helper buddy, which is another Q. And that Q is just the wingman who's just going to carry that 1 electron, just hold on to it. Hey, like, can you hold on to my phone or something really quick? I have to go to the bathroom. I don't know. It's just going to hold on to that 1 electron. And then in the second step, another coenzyme Q reduced will come in, will do the same thing, pump 2 protons, reduce a second equivalent of cytochrome C, hand its other electron to the Q minus, and the Q minus will then have 2 electrons and it'll gain 2 protons to balance charge and will become a second coenzyme Q reduced. So the net is still 1 coenzyme Q reduced, reducing 2 cytochrome Cs, pumping 4 protons. So this is like no net gain or loss of coenzyme Q reduced. So does that make sense? Anything I can clarify there?
So technically complex 3 has like 2 transmembrane proteins. Like it's, I guess what I'm asking is like,
this is the same
complex
protein.
3 is like 2 complexes?
This is supposed to be, this is like step 1 and step 2, it's the same protein, which is, it's just like shown twice, like for different points in time.
Yep.
Which is,
that makes sense.
Yeah, which is why, by the way, when this Q, because it can only give 1 electron to cytochrome C, this Q became Q minus, and this is the same Q minus. All right, cool. So that's complex 3. We got through it. Let's talk about cytochrome C now. So cytochrome C is the second mobile electron carrier in the electron transport chain. This is what cytochrome C looks like, courtesy of Wikipedia. And what do we think this is in the middle of cytochrome C? What does this look like?
Heme group or
Heme, yeah. So cytochrome C's job is to transfer 1 electron per cytochrome C from complex 3 to complex 4. What is a cytochrome? A cytochrome is a protein with at least 1 heme residue. What's another protein that has a heme? Hemoglobin, yep. So this is the heme center of cytochrome C. So whereas hemoglobin uses its heme and its iron to simply coordinate oxygen at the lungs, bring it over to the tissues that are exercising or are active and are using aerobic respiration, they're utilizing O2, they're producing CO2. So whereas hemoglobin's job is to just coordinate oxygen with the iron heme and then lose it later, cytochromes will use iron in redox reactions. So as we saw, cytochrome C gets oxidized and reduced, so that's the iron's job there. There's another example of cytochromes that the AEMC seems to like. Does anybody know another example of a cytochrome?
Is myoglobin a cytochrome?
Good guess, but it's more analogous to hemoglobin. Its job is to hold on to oxygen for the muscles so that when the muscles are really active, they have like a backup oxygen storage.
It also, has a
Anybody-
heme, it also has heme though, right?
Yep. Yeah, so it coordinates oxygen in the same fashion as hemoglobin. It just is a 1 subunit protein that doesn't have cooperativity.
Gotcha.
Anybody heard of cytochrome P450s? Cytochrome P450s. So cytochrome P,
You're saying 450?
yep, 450, yep.
Okay.
450, yeah. Cytochrome P450s are another example of cytochromes. These are monooxygenase enzymes. And in other words, they oxidize their substrates. So cytochrome P450s are mainly active in the liver. We use cytochrome P450s mainly for the beginning of metabolism of drugs and toxins. So they'll start metabolism of drugs and toxins by oxidizing certain parts of the molecule. And until they are less toxic, such that they can just then be excreted by the kidneys. Oxidation will also often serve to increase solubility of a molecule. So cytochrome P450s will also begin metabolism of steroids. So actually, remember that who synthesizes bile? Oh, that's
Liver.
Yeah, the liver synthesizes bile. So cytochrome P450s will convert steroids that we don't need anymore into bile salts. So you look at the structure of bile salt, it's literally a steroid ring, four-membered ring structure, or 4 tetracyclic ring structure with like some extra hydrophobic groups on it. So it's amphipathic. So that's where we get bile salts from, is from cytochrome P450s in liver oxidizing steroids that we don't need anymore. And then eventually, after the bile salts have done their job, we'll oxidize them further, we'll phosphorylate them or carboxylate or hydroxylate them to make them really water soluble, and then they can finally be excreted by the kidneys. A little background on cytochromes and cytochrome C, connecting it to this topic that AMC likes, which is cytochrome P450s. Any questions here? Cool. So we're almost done. We have about like 4 more slides here. Cytochrome C oxidase, complex 4. So cytochrome C oxidase's job is to oxidize cytochrome C, pump 4 protons into the intermembrane space. Actually, we have more than 4 slides. I lied. We have like 8. And reduce oxygen into water. This is the last complex of the electron transport chain, where we finally have the terminal electron acceptor, oxygen, taking the electrons from our food molecules, our glucoses and our fats, taking the electrons from them, producing water. This is the whole thing which explains why the citric acid cycle, the pyruvate dehydrogenase complex, the electron transport chain can only be active in aerobic conditions, is because we need to eventually give our electrons over to oxygen. So the net reaction here, remember the 2 versus 4 protons. So this is a diagram that shows 4 protons. Now we're not going to get confused. 4 cytochrome Cs will get oxidized per O2 molecule because we have to go from an oxidation state in oxygen of 0, but there's 2, to -2. So we need a total of 4 electrons to reduce that oxygen to water, as well as 4 protons. And then 4 protons will get pumped into the intermembrane space per O2 reduction. Also, so note that as shown, this would actually require 2 NADH and or FADH2. This would require 2 electron carriers because each of them is only going to give 2 electrons to the electron transport chain and would only be able to produce a half O2 molecule. So this is sort of the discrepancy between some diagrams you might see where this complex is pumping 4 protons and receiving 4 electrons. The purpose of having figures that show the 4 as opposed to the 2 is that it takes 4 electrons to reduce a single oxygen molecule. So this is from the perspective of oxygen, if that makes sense. Whereas from the perspective of the electron carriers, it actually requires 2 electron carriers for a total of 4 electrons. Does that make sense? Any questions? Sweet. Yeah, so yeah, don't get confused when you see some would have 2 and some would have 4. All right, back to our overview. So again, here's complex 4 with a half oxygen molecule, 2 electrons. This would be 2 cytochrome Cs and only 2 protons. So in totality, NADH will enter the electron transport chain of complex 1, which will pump 4 protons. Coenzyme Q, 4 more protons at complex 3, so a total of 8 protons. And then lastly, 2 more protons for a total of 10 protons per NADH pumped into the intermembrane space. FADH2, whether it's via glycerol shuttle or via complex 2, the electron transport chain, FADH2 will send 2 electrons here. It'll miss out on these 4 protons, but it'll get 4 protons from complex 3, 2 protons from complex 4 for a total of 6. Any questions here? Just kind of a summary.
Okay, I'm sorry. So complex 4 is actually it's just 2 protons, not 4. The only reason why the last slide showed 4 was because it's from the perspective of oxygen.
Yes,
okay.
Exactly. Whereas this is from the perspective of the electron carriers.
Gotcha.
Let's talk now about the last step, which is where we get all of our ATP from. So we've set up this wonderful proton gradient, which is potential energy, electrochemical potential energy. And we'll utilize ATP synthase then to couple the dissipation of the proton gradient with phosphorylation of ADP to ATP. So let's talk now about our ATP synthase, who's kind of the quarterback, something football analogy. ATP synthase is an enzyme that couples the electrochemical proton gradient with ADP phosphorylation. Don't worry about the subunits here, but protons will enter through the top and will go through the bottom here. The protons pass through ATP synthase. They cause rotation of this gamma subunit in the middle. We can think about the gamma subunit almost as like a motor. It's a motor that's powered by proton flow. And the motor produces energy in the form of ATP. We have ADP and phosphate. This arrow should be going the opposite direction. It's technically classified as a ligase. We can think of it as an ATPase running in reverse. And again, don't worry about the subunits here. Don't worry about avidin or actin filaments or histidine or nickel. Any questions?
It's a proton specific, right?
Yes, it's a proton. Its channel is proton specific. So we can imagine it's probably very small. And would it be lined with acidic or basic residues, maybe?
Acidic.
Acidic, right? Which would be negatively charged, which help conduct protons. Perfect. I don't know if that's 100%. I don't know if that's true, but sounds good. If you had an MCAT question, you would guess acidic residues. So let's look at what's happening at the bottom in the part where we're actually making ATP. So there are three sort of binding sites here. We have an ATP binding site. We have an empty site where ATP leaves from. and we have a site that has ADP and phosphate. So when these protons enter into the enter through the complex here, the ATP synthase, the protons will cause rotation of the gamma subunit, which will cause conformational changes in the subunits. So if we start here, we have ADP and phosphate. When H plus comes in, we'll then like squeeze them together, make ATP. This ATP that was in this binding site will leave, and a new ADP and phosphate will come into the third site, the empty pocket, ready to be phosphorylated. Another 3 protons come in, we rotate the units, we do a conformational change, we squeeze ADP and phosphate together to get ATP, we phosphorylate ADP, ATP. ATP leaves from that binding pocket, and a new couple come in, and so on and so forth. Any questions here? So don't super worry about these subunits. We're just showing details so that everything sticks better. This will be important for understanding the yield of ATP. Does anybody remember like learning in biochem that that the total oxidation of a glucose molecule was 36 to 38 ATP, or like 36?
Yes.
Yeah. Did you ever wonder why they changed numbers on us?
Because they found out that it was that I believe they found out about FADH2, how it's not equal to NADH and how much ATP is produced.
That's going to be one part of the puzzle, and that's going to explain the like range of like 36 to 38, or the range of like 30 to 32. We'll come back to that.
I have
There's
a question actually before we move deeper into the topic. Sorry, I didn't bring it up before, but on the last slide, what type of view was that? Was that like a cross-sectional?
cross-sectional.
Okay, because I was just confused at how it would look like that. Okay, thanks. That answers it.
Exactly. Yeah. So we're looking at like, we sliced it open and we're looking inside this. Yeah.
Okay, thank you.
So that the FADH2, we'll come back to that. It's going to be one component. The other component is that previous calculations weren't taking into account the phosphate translocase. So let's talk about these 3 types of transporters. So we have one transporter for getting mitochondrial matrix ATP back to the intramembrane space, where it can then go to the cytosol through the pores of the outer membrane. So that's called adenine nucleotide translocase. And ADP and ATP are going to exchange in this protein via antiport. So we can imagine that ATP concentration would be higher in the matrix because that's where we're making ATP. And ADP concentration would be higher in the intramembrane space because that's where our ATP that we'd already used would be coming in from. So that's the first transporter. The second transporter, of course, is ATP synthase, so H plus will go in here. The synthase will combine adenine diphosphate with phosphate to make ATP. The last transporter, and this one, remember, this takes 3 H plus, there should be a 3 here for clarity. The last one is phosphate translocase. So this guy is actually a symporter, which symports H plus and phosphate to get phosphate into the matrix. Particularly because right now the intramembrane space is high in plus charge, so it's harder to get a negatively charged molecule into the matrix. So the H plus gradient fuels phosphate translocation into the matrix. So even though ATP synthase only needs 3 H plus to make 1 ATP, a 4th H plus is necessary to actually get phosphate to this guy on the matrix side. So accounting for this is one of the reasons why that old 36 to 38 ATP yield of glucose was lowered to 30 to 32 ATP. So because now we're accounting for the fact that some of the H plus gradient is required to actually get phosphate into the matrix. Does that make sense?
It does, yeah. Should we be using the 4H plus equals 1 ATP or the 3H plus equals 1 ATP?
Context dependent. I don't even know if you would need to know stoichiometric ratios for H plus gradient versus ATP production. Context dependent, the MCAT would have to account for the nuance.
Okay.
So if, for instance, if I don't know, if they wouldn't ask you a question, but if they did ask you a question and they had not mentioned phosphate translocase, you would choose 3 or 4, but they wouldn't give you both options. Does that make sense?
Gotcha. Yeah. All right.
Sweet. So energy yield summary. NADH oxidation, we said, causes 10 protons to be pumped into the intermembrane space. We said there's 4 protons per ATP produced if we account for phosphate translocation. So we take our 10 H plus, we divide by 4 H plus per ATP generated, we get 2.5. So that's where our number for NADH comes from, 2.5. Used to be 3, but they weren't accounting for phosphate. We said FADH2 oxidation causes 6 H plus to be pumped into the intermembrane space. And if we take our number of 4H plus per ATP, we get 1.5 ATP per FADH2. So those are where our 2.5 and our 1.5 numbers come from, because FADH2 misses out on the 4 protons from complex 1. We've been mentioning the H plus gradient as an electrochemical gradient. The chemical gradient is that there's a lower pH in the intermembrane space in the matrix. And the electrical gradient is that there's more positive charge in the intermembrane space than in the matrix. You kind of think about it like a capacitor, where ATP synthase is like the wire that you connect to discharge a capacitor. Questions here? Cool. So we have reviewed the electron transport chain. Let's introduce an interesting example that I alluded to earlier. Back in like the 60s, there was this protein that was found in brown adipose tissue. And at that point, they just called it uncoupling protein before they had discovered other uncoupling proteins. So now it's called thermogenin. He said one of the purposes of mitochondria is to generate heat in adipose tissue to protect vital organs from cold temperature. So what would happen if we inserted a proton conducting channel into the inner membrane? So now H plus has 2 options. Which would it rather go through?
It would rather be in equilibrium, so it's not going to form a strong gradient, right? Where there's so much H plus being pumped outward.
Yeah, so we'll assume that the electron transport chain is still just doing a satisfactory job of getting H plus into the intermembrane space. But if you're an H plus proton, you're thinking from the perspective of a proton, and you're like, do I want to take the toll road where I have to like contribute to ATP synthesis, or do I want to take the freeway? Which way are you going to go?
Freeway, of course.
Take the freeway, obviously. You're going to go down the path of least resistance. Nobody wants to pay, which is a toll road, $6. I don't know. They don't have a lot of them here in my part of California. Oh, we've got our mascots going to come and check in with us pretty soon now. So we have this. So this will result in uncoupling. Remember, we couple proton gradient dissipation with ATP synthesis. This will actually uncouple those two, so that we will just have protons going through the uncoupling protein, the thermogenin. And then, but when we dissipate this energy gradient, this potential energy gradient, when we dissipate it without making ATP, there's actually energy that has to, from the gradient, that has to go somewhere. Because of conservation of energy, we know that energy has to go somewhere. Hi, buddy. Yes. Scratch. And that energy will become heat. It will be lost in the form of heat. So this is how mitochondria in the brown adipose tissue. Want to say hi to everybody? Oh, scratches. Say hi. Say hi, son. So thermogenin is this protein that's expressed in brown adipose tissue. And what happens is in cold temperatures, the adipose tissue will insert thermogenin into the inner membrane of the mitochondria and will just release the proton gradient in the form of heat. Some questions here?
So does, how do I ask this question? Brown adipose tissue, does it still have ATP synthase, or is it replaced by UCP?
It's got both, but the protons, they want to go through the path of least resistance. So it's got both, but then in cold conditions, uncoupling protein gets expressed, it inserts into the membrane, and the protons will go through there instead.
Gotcha.
And uncoupled proteins are only present in adipose cells, right? Adipocytes.
Correct. Yeah. Now there are certain chemicals which will do the same thing. Like one example is 2,4-dinitrophenol. There are chemicals that'll do the same thing, where they will lead to forming a channel that protons can simply flow through for free. And when those get into our cells, that is unregulated dissipation of the proton gradient. We lose out on a ton of ATP, and this is actually very life-threatening and actually can be irreversible too. So yeah, bad if a chemical comes in and does this in willy-nilly in just random cells and tissues, but good if we're regulating this and we're only doing it in certain cells and to generate heat.
Is this what's going on when we shiver, or is that something completely different?
So there's like 4 main methods that humans will like protect themselves from cold. So one is like shivering, so our muscles will like quake and that'll generate energy. There's also well, we don't actually really use this well. Like my cat can use this other way well, which is like raising the hairs on, because like in mammals that have hair, lots of hair, we call it fur. So like, you ever hear about like if you're in cold temperatures, like really, really cold, you can like stuff newspaper into your coat to stay warm?
No, I can't say I have.
Yeah, I mean, so unfortunately, like this example comes from like people who are homeless, and they will, like one way that you can deal with like being left out in the cold, like a really cold city or something, could be to stuff a bunch of newspaper into your coat. And this leads to a lot of air being present, and the air can actually act as an insulator. So mammals that have fur can do a similar thing where they elevate their hairs, and that stores a bunch of air inside of the hair, which serves as an insulator.
It's like mini convection or something, right?
Yeah, kind of like, well, it's like it's adding like another layer that heat would have to be convected through. Yeah. So it's making it harder for heat to leave because of the insulating air. So that's method number 2. So shivering, hair raising, that doesn't really help us because we don't have as much hair as like my cat does. Method number 3 would be huddling. So we'll decrease our surface area so that we're losing less heat due to convection as well. And then this could be another method. We also can raise our basal metabolic rate as well. And then this would be another method, would be thermogenin. That's why they call it non-shivering thermogenesis. Cool. Sweet. And then I think this is our second to last slide. So this one is describing the bioenergetics of the electron transport chain. So on the left, we have 2 axes. On the left side, we have free energy going from high to low. On the right side, we have, I don't like that they call it redox potential, because like, what is it, reduction potential or an oxidation potential? It's a reduction potential. So oxygen has the highest reduction potential here. NADH has the highest oxidation potential. And it also has the highest free energy. If we look at the values, they're on this chart, but I looked them up. If we look at the values for the oxidation potential of NADH versus FADH2, NADH has an oxidation potential of plus 0.32, whereas FADH2 has an oxidation potential of 0.22. So that helps us start to explain why NADH is able to enter the electron transport chain earlier because it has a higher oxidation potential than FADH2. And remember, this stems all the way back to the different types of bonds that, when oxidized, will lead to reduction of electron carriers. Carbon-to-carbon bonds, which are less polar, when oxidized, will reduce FADH2. Whereas carbon to oxygen or carbon to sulfur, as we saw in glycolysis in the citric acid cycle, when oxidized, will be a higher, more polar bond, and therefore a higher reduction potential will lead to, will be a higher oxidation potential and will reduce a better electron carrier, aka NADH. So the potential energy from these electron carriers is converted into electrochemical energy. So we're converting our potential energy. This is all spontaneous because we're going downhill, we're losing free energy. But we're saving some of the free energy in the form of a proton gradient. Now, I think it's only like 33% of the energy of the redox energy. Like only like 33% actually gets converted into useful proton gradient, but it's a lot better than like I think an internal combustion engine, for instance, like in a car, is like a lot worse than that. Cool. Any questions on this slide? Finally, let's talk about the total ATP production from one molecule of glucose. So glycolysis, what are the energetic byproducts of glycolysis? What do we make? We make an NADH. How many NADH?
2 NADH, 2 ATP.
Yeah, we make a net of 2 NADH and 2 ATP. And our NADH, remember, are good. Yeah, pyruvate, right. We make 2.5 ATP per NADH, so we get 5 ATP from the electron transport chain and oxidative phosphorylation. And remember, in the glycerol shuttle, which is present in brain and skeletal muscle, I don't know if I mentioned that previously, with the glycerol shuttle, for cytosolic NADH, which comes from glycolysis, complex 1 gets bypassed. So in those types of tissues, NADH acts like FADH2 and generates only 1.5 ATP each for a total of 3 ATP as opposed to 5. This results in 30, as we'll see, ATP per glucose in those tissues instead of the 32. So if you ever saw 30 to 32 ATP and were confused, which I was certainly lots of times, this goes to explain why there's like the 30 to 32 range. So depending on the tissue type, and therefore depending on whether it uses the malate shuttle, which is no net loss or gain of NADH, you get 32. But if you use the glycerol shuttle, you would get 30 because your NADH are therefore acting like FADH2. Does that make sense? And then pyruvate dehydrogenase is going to generate how many NADH per glucose? 2 NADH, so we get another 5 ATP. Citric acid cycle, what do we get from there? How many NADH per glucose?
Is it 3?
This is 3 per round of citric acid cycle, but remember 2 acetyl-CoAs, because of 2 pyruvate, will go from 1 glucose. So total of
6.
Total of 6 NADH, giving us 15 ATP. And then we'll also get 2 FADH2 per ATP, 1 per round. Will give us another 3 ATP, because 1.5, and then 2 GTP, which are 2 ATP equivalents. So if we add it all up, we get, let's see, 15 plus 10, 25, plus 5, 30, plus 2, 32 ATP per glucose molecule. Any questions on the calculations? Cool. All right. So this is our last slide of the day. We're going to head on over to, so next week, we're going to be starting with fatty acid catabolism. I'll just like skim through these slides. You can see we'll be doing like lipolysis and lipogenesis, fatty acid catabolism, so activation of fatty acids, translocation, and then we will also cover in next week's lecture beta oxidation and fatty acid synthesis. Any last minute questions while the recording's still going?
I'm just a little confused about intermembrane space has more ATP. Should it have more ADP, or is it because ATP is being pumped back, or hydrogen is being pumped back and then it's okay? Yeah.
So matrix is where we're making ATP, right? And then intermembrane space is, so we're going to make ATP in the matrix. We're going to send it over to the intermembrane space, which will then go to the cytosol. And the intermembrane space, aka the cytoplasm, is deficient in ATP, but it is abundant in ADP. So therefore, that's why we can use these guys in an antiport fashion. Did that answer your question?
Yes. Okay. Thank you.
Awesome. Okay, any other questions while the recording is still going? All right. I'll see everybody on YouTube next week, and thank you for your participation and your attention.